Hello, 205ers. I made this video just to talk about the Unit 1 labs, so we'll go over them. Lab 1 is the microscope lab. I'll probably already turned that in. It's pretty much self-explanatory. The only thing I would say is if you're actually in a lab and you find that you can't see your specimen, a couple things you can check. This is called the nose piece. I like to call it the turret because like a gun turret. But make sure that that's clicked in place because sometimes students will not be able to see the specimen here, but really the objectives are just off a little bit, so they're not clicked in. The condenser, I think you can use that little lever to switch, to move back and forth to get more or less light. And then a couple of terms they talked about with the microscope. Resolution, that's the ability to distinguish 2 points as 2 separate points. So when you're looking in the microscope, if it has a high resolution, if it's a high resolution microscope, you'll have the slide here, and you'll be looking at 2 structures that may be really close to each other. If it's a low resolution microscope, you won't be able to tell that those 2 points are 2 distinct points. If it's a high resolution, you'll be able to see each one of those points. And then parfocal, the microscopes at Pima are parfocal. That means the image stays in focus when moving from one objective to another. So usually you start to focus with the lowest objective, and you use the coarse adjustment to move the stage up and down. And then when you go from, say, the 4x to the 10x, it should be pretty much in focus. You just want to use the fine adjustment knob to get it in focus for your eyes. Okay, so that was lab 1. This is lab 2, preparation of bacterial smears and simple staining. So in this part of the cartoon, well, we're making a smear. This cartoon is making a smear on a slide from a broth culture. So here you are. You want to mix that broth culture. You can put it on a little mini vortex or two, or you can just swirl it around, mix it back and forth. And you do that in your non-dominant hand. And then you get your inoculating loop here in your dominant hand, and here's our Bunsen burner, and you put the inoculating loop in. You got to find the right angle so the tip of the loop here glows red, and then you have to, before you put it into your broth culture here with the microbes in there, you have to let it cool down a little bit, and then you take your little finger here and unscrew this lid. And then your non-dominant hand, you want to sterilize the tip of the culture tube over the flame. And then you can go in with your inoculating loop and swirl around a little bit and get some microbes in your inoculating loop. Notice you still have the cap in your hand here. Oh, you should also have gloves on. They don't have gloves in this cartoon, but that's okay. And then you want to sterilize the tip of your culture tube again, because if you're trying to, especially if you're trying to save these microbes in this broth, you want to make sure that this stays sterile. So you sterilize the tip of your broth culture, and then you put the lid back on, and then you take your microbes in the inoculating loop and you smear them on the slide. And then you re-incinerate your inoculating loop, and then you can put your inoculating loop down. So this is making a smear from a broth culture. So you just drop a loop full of microbes from a broth culture on a glass slide, and then you dry it, and then you heat fix it. You can also make a smear from an agar slat culture or an agar petri dish. So in this cartoon, it's an agar petri dish. These are colonies, so these colonies could contain millions of microbes. So what you want to do is you want to put a little drop of water, it can just be non-sterile tap water, here on the glass slide, and then you go in and you sterilize everything as usual, but you go in and you just get a little loopful of microbes from your colony, and then you swish it around in the water here, and then you let it dry, and then you heat fix it, and then you'll go on to stain it. And a colony is just, it's a visible mass of cells. So this thing here is a colony. It's a visible mass of cells. It's grown on a solid media, and this colony originally came from a single cell and grew into that giant colony. Okay, and then after you've smeared your slide, you want to stain the smear. So the simple staining procedure. You can put your slides, or all these slides, because a lot of times you'll have to share with your bench partner. You can put these slides on a rack. These slides that have been smeared and are ready to be stained, you put them on the rack, and you can, in a simple stain, you just cover with a couple drops of crystal violet. Don't flood it, and you want to wait 30 seconds, and then you hold the slide over the rack at a 45-degree angle and just rinse gently. And usually, I think in the lab we have DI water for that, because they've got a little water dropper here. And then you want to blot your slide between the pages of this bilbulous paper. Now sometimes people will take the paper that you use to wipe the lenses off, which is really expensive paper. So if you're in the lab, you want to make sure that you get this bilbulous paper, and then you can see the person here is putting that slide in the bilbulous paper and then just blotting it. And now you're ready to look at it in a microscope. Okay, in your lab too, they talked about this PEA and DLA media. So if you've got a sample and you don't know if you have gram-positive or gram-negative cells to start, you can grow your sample, like from a broth culture. You can grow that on PEA, and PEA will only grow gram-positive cells, so that you'll only have gram-positive cells on PEA media. And then if you use DLA media, that only grows gram-negative cells. So in the gram stain, you make a smear on the slide from the bacterial colony. So that means you would get your smear sample from a plate, an agar plate, and from a colony. And then you let the slide air dry, and then you heat fix it. Then you start the staining. And first you apply the crystal violet, and then you can see that all the cells are stained purple at this point, because that crystal violet is staining the peptidoglycan in the cell wall. And then you apply this iodine, which is a mordant that kind of locks the stain into the peptidoglycan cell wall. And then they're still all purple. You can see here from the crystal violet. And then you use this alcohol to de-stain or decolorize. And you can see that the gram-positive cells kept the purple stain, and then these gram-negative rods lost that purple stain with the decolorization step. And then you apply safranin, which is a counter stain, and then the gram-negative cells will take up this pink safranin stain pink, and the gram-positive cells will stay purple. So a few tips. We just kind of mentioned this. The gram-positive cell walls have a thicker peptidoglycan, so it retains that crystal violet primary stain. And we've talked about this. The iodine keeps the crystal violet in the cell wall. It's called a mordant. That's what mordants do. Decolorizing is a critical step in a gram stain, because you can decolorize with the alcohol too much or too little, so you want to use the right amount and the right amount of time. And then you want to use fresh cultures of bacteria to do a gram stain, because bacteria, as they get older, they don't retain that crystal violet stain, and then you can't tell if it's gram-negative or gram-positive. Lab 3, just Nutrient Agar. There's 3 ways you can do Nutrient Agar. So you can do a Nutrient Agar plate. So this is agar mixed with nutrients, your broth mixed with your broth. You can do a Nutrient Agar slant in a tube. So this is Nutrient Agar, but the bacteria are growing on a slant culture with the Nutrient Agar broth. And then just plain Nutrient Agar broth, bacteria can grow in that without any agar growing on any agar slant. Okay, next one from lab 3 is the streak plate. So in this cartoon, doesn't show it upstream from this, but you could maybe get a loop full of bacteria from a colony. So let's say you have a plate up here and you've got all these colonies growing. You can, sterilize your loop and get a loop full of bacteria from a colony, and then you do this this dilution. So you're diluting the microbe out such that when you see the result, each single microbe will result in a single distinct colony that you can actually characterize. So you can talk about its morphology, etc. So these are not single cells, these are colonies of cells. So the first loop full, you come in and you streak back and forth, but this is going to be too thick. So this is number 1 over here. This is number 1. It's too thick. You can't see the individual colonies. And then what you do, you have to re-sterilize your loop, and then you go back in and you pull down some of the microbes in this second dilution. But again, that second dilution, it's still the colonies are too close together to see. So then you re-sterilize your loop and you come in and you pull down some original cells in this third, and it's getting a little better here in the third one, but you still can't see the colonies very distinctly. But then check it out, when you take this fourth one, you pull some of the microbes from here and you streak them out, and you can really see the colonies well, and then you can go on to describe the colonies, and you can tell, and you can do more biochemical tests, but then you can see which microbe you have. So in the lab, you take a sterile inoculating loop, you move it across the surface of a culture dish. So basically you're thinning out the sample, and you're isolating individual colony forming units, which usually could be a single cell or a couple of cells, and they grow into colonies. That's the streak plate technique. And the inoculum is just what you've inoculated your loop with. So the inoculum is the microbes in the loop. It's gradually diluted to a point where after you incubate for, I don't know, maybe you could incubate that for a couple days, you get these colonies from a single colony forming unit. And then now, like I said, you can examine the colony morphology. And we'll look at the cartoon for colony morphology in a minute. But this is lab 4, isolation of cultures from natural sources. So this is a fun lab to do when you're in the class, because you get to kind of go on a bug hunt, and students can get samples from their cell phones, doorknobs. Students have gone to the toilet seats, so you can go anywhere when you're on campus. But anyway, so then you actually use like a cotton swab, and it's a sterile cotton swab, and you swab your surface of interest, and then you take that swab, and it's like you're painting back and forth on this. So you're trying to get all the microbes as thin as possible so that they can grow into these single colonies, and then you can take the colony that you think looks interesting and further investigate it on another plate, on a streak plate. So when you get your colonies from your natural sources, so for example the cell phone, you want to select the colony that's isolated from its neighbors. So let's say this one. This one looks interesting here. Or you could use this one. This looks kind of huge. That is isolated from its neighbors, so you can do a streak plate for further analysis. Because we're only interested in one organism to characterize in this lab, and we want to identify it in terms of its single cell characteristics and its colony morphology. And then this is just a cartoon of the streak plate. So this inoculating loop would go here. You'd take just a few microbes. Microbes should all have descended from a single microbe, so they should all be the same. So you go in with your inoculating loop and take a loop full from here, and then you would put it on this streak plate, and then you'd be able to identify the colony. And then here's how you describe colony morphology. So the shape of the colony could be circular, rhizoid, irregular, filamentous, or spindle. What's the margin like? So you're looking at a colony, they're usually circular, but is its margin entire, or does its margin look like this, filamentous? How elevated is it? So you kind of have to look at the plate on the side. Is it a really small colony, or is it a large one? It's kind of hard to tell the texture. The appearance, most of the time it's dull, but sometimes it can be shiny. And then is it pigmented? So is the colony purple, red, or yellow, or is it non-pigmented? Is it cream, tan, or white? And then is it opaque? The optical property, can you see through it, or is it, or can you not see through it? So this is a Serratia marcescens colony, and it defines its characteristics of the colony. So remember, this is not a single cell, this is a colony, so it could be hundreds of thousands of cells in this colony. And then it gives the characteristics of that colony. So this is where a lot of students have a hard time remembering the difference. You use a smear to describe the cell morphology. So in a smear, when you look at this under a microscope, you're able to see single cells, and you're looking at single cell characteristics, like is it gram positive or gram negative. To describe cell morphology, you use a smear, and to describe colony morphology, you use this streak plate. I think that's it.